Hello. On behalf of the Biochemical Society and Portland Press, I'm pleased to welcome you to today's webinar, which is part of our Biochemistry Focus webinar series. Topics in the series include different research areas in the molecular biosciences, as well as practical sessions to support career development. Each webinar will give you the opportunity to ask questions via text, and we welcome suggestions for future topics and speakers to feature in our webinar series. Please see the website for details. Hi, my name is Catherine Jopling. I'm the chair of the Biochemical Society Genes panel, and so I'm here to introduce today's speaker. I'm an associate professor at the University of Nottingham with an interest in RNA biology. So in this webinar, we're going to hear from Dr. Fredericus van Eeden. He's from the University of Sheffield, and he will discuss the theoretical and practical aspects of CRISPR modification in zebrafish. Just to introduce his work, zebrafish is a widely used model organism spanning the fields of developmental biology, epigenetics, transcription, and more. The advent of CRISPR technology for making precise modifications to the genome has surpassed siRNA and talon technologies in many applications. Much work and outreach has focused on genome editing in mammalian systems. However, vertebrate model systems such as Zebrafish and Xenopus are used by multiple vibrant research communities who have adopted CRISPR technology for their own purposes. Dr. Fredericus van Eeden is a senior lecturer in the School of Biosciences, and his research interests include using the zebrafish as a genetic tool to study development and disease. So just before Fredericus gets started on his webinar, to mention that questions will be asked after the presentation. So in the meantime, please type your questions into the question box, which you can do at any time during the talk. When it gets to the end, then I will try to arrange for Fredericus to answer as many questions as the time allows. Okay, so that's it for the introduction. So I'm going to hand over to Fredericus now, who's getting his slides up on the screen, and I'll see you for the Q&A session at the end. Over to you, Fredericus.
Okay, thank you. It's a pleasure to be here. I'm just seeing whether this is also in view, so I'm just going to move this out of sight a little bit. This is the first time I do a seminar like this, although we are doing these in the university, and it is because many people are working on different systems, and they're kind of keen to get into the zebrafish, especially since now it's so easily possible to make knockouts in the zebrafish using CRISPR technology. And actually also further genome modifications, although this is a bit more, still a bit more work, so to say. It's not as well established. But it may be good to simply give a kind of a practical guide of how we do this, because we have made mistakes and we have learned things along the way, and I think it's good to maybe share how we go about. And I thought I'd do this simply by kind of using a hypothetical scenario, and I hope that is a good idea. So the scenario that I would do is, you have identified a human or a mouse gene of interest, and you would like to create a zebrafish knockout. So what do you do next? Where do you really start? Well, the first thing you need to do is, if you work in a different organism, is that you have to identify the zebrafish homologs of your gene. And then you need to design a CRISPR. So what we use for identifying homologs is basically, out of convenience, we tend to use Ensemble. And Ensemble is a website, I hope you're aware of this, where you can find your gene of interest relatively easily. So here we went for a human gene. So I kind of chose the POW2F1 gene. This is because we've used it in several practicals. And I found the human gene on Ensemble, as you can see over here. If we want to then go to the zebrafish homologs of these, it is very advisable to look at the gene tree, and I will show you that. In addition, we look at another place, and that is a list of orthologs, and these can all be found in Ensemble if you look for the gene of interest. So if we click on the gene tree, what you will get is a tree that roughly looks like this. And you can navigate this by clicking on some of the buttons over here, and that will allow you to find the homologs of the human gene, which is located over here. And you can see it nicely in a phylogenetic tree with the zebrafish gene. And this is really important because the zebrafish has underwent a partial genome duplication. And often where you think there is just one gene, it turns out that if you look in detail in the sequences, you can see another gene over here, which doesn't have a very intelligible name, but nevertheless, it is an ortholog of the POW2F1 gene in humans. So if you want to create a knockout, you better prepare to make a knockout for both this gene and this gene, because they're both POW1, they're both homologs, basically. If you look towards the orthologs, you can also see that there is not a clear relationship between the zebrafish and the human, because it says over here, if you look at this list, which you get if you click on there, if you look at human, you get a one-to-one or a one-to-many relationship with zebrafish. So this is an important piece of advice when you start such a knockout project. Be very clear that you have, that you know of all the zebrafish orthologs that might be present. You might, you can easily miss one. Okay, once you have identified one of the genes that you will knock out, you can simply click on this gene, and then you will get to the gene landing page for the zebrafish. So it's very convenient clicking through the organisms. And then if you go to the transcripts that you can see, you need to select the transcript that you want to use for creating a knockout. And what we usually choose is the proteins that are the longest, and there's a tag over here which is very useful that says ensemble canonical. That means that this is the most common transcript variant that is present in the zebrafish. And that's usually what we use as a start. And then when you want to design your CRISPRs, you need to choose your target area very wisely. You might say, and we have made some very painful experiences with this, you might say, okay, I'm really going to make my CRISPR mutant really early in the gene, because then we are sure that everything else behind it is going to be destroyed and knocked out. However, this is often not the case. If you make a frame shift mutation really early in a gene, often the ribosome simply finds the next ATG molecule or the next ATG codon and will start forming the protein again. So you have to be careful with this. Of course, if you make a CRISPR that is very far in the protein, then you will have some active components still being present. And in order to look at where the important parts of the protein are, we simply in Ensemble can click on the protein, and this will bring up this kind of protein analysis where several conservation detection programs are used to identify the important parts of the protein. And for POW2F1, which we're using as an example over here, you can clearly see there's something very conserved over here, a POW-specific domain and a homeobox domain. So it's probably going to be a transcription factor. And that means that if you make your mutation, your frame shift mutations before the actual active domain, then you will be in business. Another important thing is to look at splice variants, and this can also be done simply on the landing page for the gene in zebrafish, and there you will get these different transcripts which you see over here. You can recognize them over here, and you can see that there are certain exons, as you can see, these ones and these ones, that are specific for certain isoforms or splice variants of the gene. And of course, if you want to create a knockout, you don't want to have this isoform knocked out, but not that isoform knocked out. So when you decide where you want to place your CRISPR mutation, try to select exons that are common to all of the splice variants that are shown. So I would normally go for, you know, exon 3 or 4 in this case. Right. So where do you go next? Once you have identified the transcript that you want to use to identify your CRISPRs and roughly the exons where you want to do this, then you can take this number over here. You have to remove the .3, I think, but you can take this number over here and then copy it into the ChopChop website. So this is the website that we tend to use to design our CRISPR guides. It is basically out of convenience. We find this one the most convenient. There are a few other ones. You know, there are other sources where you can see guides. So there's a if you're familiar with the UCSC genome browser, there's a Zebrafish genomic hub that you can find under the track hubs. It's called Zebrafish Genomics, made by the Burgess Lab. And if you activate this track, you can see a kind of pre-designed guides that are, you know, according to their algorithms, are likely to be active guides. However, as I said, we tend to use the ChopChop website. If you use this, there are a few things that we need to pay attention to. First of all, you need to use the right version of the genome. So there's another one which is called 10. We use 11. That's the newest version. And in the basic settings, we will set the product size, and I will come back to this later, to about 90 to 175 before I actually send off this sequence. Right. Okay, when you send off this sequence, after a while, the ChopChop website will produce a view like this one over here. And you can see different exons of the gene. And above this are all these little arrows, which are basically designed guides. And you can then scroll and zoom in on the exons that you were interested in. So you need to make a note of that, which exons you actually want to target. And then you get a view like this. And here you can see different guides that have been designed by ChopChop, and they have a kind of a scoring system where the green ones are the good ones, the red ones are the bad ones, and the orange ones are the ones in between. However, we have found that these scores from ChopChop are relatively poor predictors of in vivo efficiency. And actually, we find that many guides are perfectly active that have low scores. So when I look at these guides, I actually look at a number of things simply by eyeballing them. So I generally worry if there are long stretches of single nucleotides or dinucleotides. So something like this, I worry a little bit about it. But you know, if I have to use this guide, I wouldn't worry of ordering it. They're not that expensive anymore. I am a bit worried about self-complementarity. So for instance, here's a guide that has quite a bit of self-complementarity. I would avoid using that guide if I could. Sorry. Then there are these tables over here. This shows the number of mismatches. So this is a perfect ectopic site for the guide where it would cut in the genome. There's none over here. But there are some guide binding sites in the genome that have more than one mismatch. You can see those over here. So this is guides with 3 or guide binding sites with 3 mismatches. I generally do not worry too much about these mismatches unless they go into the hundreds. And that is because you know, we are working in zebrafish. Zebrafish can be crossed after you've injected guides. And that means that if there are off-target mutations, you can clean them away from the mutation of interest by crossing them out to wild-type lines. And zebrafish have 25 chromosomes. So that means there's only a 4% chance that any of these off-target sites you see over here will actually be on the same chromosome as where you want to place your mutation. And that would mean that it would be more difficult to separate. So in most cases, it's very easy to get rid of any off-target mutations that you might have induced in your fish after applying the CRISPR-Cas9. Okay, so this is how a CRISPR looks on the genome, how it's actually binding. So you can see here the CRISPR, the CR-RNA. Here you can see the tracer part of the guide RNA, and you can see that the CR-RNA binds to, here the strand that is opposite if you read the gene sequence over here. And here you can see the PAM site, which is an essential sequence for the guide to work. And it's basically, it consists of 2 Gs if you read in this direction. Now, what we do if we are on the ChopChop website is we just look through the guides and we find guides that disrupt restriction sites. And ideally, we select enzymes, restriction enzymes that cut directly into PCR buffer. And if you want to know which ones that are, you can look on this website over here. If you need to use a guide that has no restriction enzymes, that is also possible. I will come back to this later. Anyway, so you click on one of these guides over here, and then ChopChop will bring up the next window, which is this one over here. It shows the guide on the sequence. And what you can see are over here are a number of primers that have been designed to allow amplification of the region where the guide is actually going to work. And these are going to be handy to determine whether the guide is actually doing what it's supposed to do when you inject them into zebrafish embryos. And when we evaluate a guide, what we do is we look at the restriction sites that are also annotated in this particular view. You can hover over these blocks over here and see which restriction is, or which restriction enzyme is actually is detected over there. And that will mean, so if you find, for instance, a BSL1 site is a fantastic enzyme because it has 2 Cs over here. Then it has a number of undetermined nucleotides and then 2 Gs. That's the recognition site of BSL. So it means that if you have a guide that will cut over here and will create insertions and deletions at this particular site, this will destroy the restriction site, and you will be able to see this and evaluate this on a gel, very soon after you've done your injections. You can also, as I said, you get this list of primers that you can use to, you can order these primers along, and you can use those then to evaluate whether your guide is working. Okay, so when you've chosen a certain guide, you can order them. And then the next thing is basically you inject this guide into zebrafish embryos. And I've put the, the pipetting scheme over here because I always like it when people publish this. That's very easy for me, for you to copy. So this is what we pipette together. And I will come back to some details of this later. We inject the zebrafish when the eggs are freshly laid. Then we wait for 24 hours. We do a hotshot DNA isolation. It's a very quick and dirty isolation, and it only works on relatively short PCR products. So that is why I said that we need to set our PCR products to a relatively short size when we go into the ChopChop website. Then when we have isolated this DNA, we do a PCR with the primers that we have ordered over here, and we do a restriction digest, and we can do this restriction digest then directly in the PCR buffer, so everything is very convenient in this respect. And then you get your gels, you run your gels, and you can see whether your guide is working. So here you can see some uninjected embryos. We do the PCR. When it's not digested, you get a straight band. However, if you digest your PCR product, you can see that you get a nice digestion product, and you can see some different examples over here. Here we have embryos that have been injected with the guide. Now the guide cuts the DNA, and as a result of this cutting, the DNA repair mechanism will lead to insertions and deletions. And these insertions and deletions will destroy the restriction site that we use to digest the DNA. And as you can see over here, we have digested DNA and undigested DNA, and we can see that all the PCR product, there's almost no digestion taking place anymore in comparison to the undigested ones. So that means that the CRISPR that we have selected is actually working, and it's creating mutations at the site of interest. And if this looks okay, then we'll take a parallel batch of fish that we haven't isolated DNA from, and we raise the fish that are that have been injected. So we raise the injected embryos. These embryos are mosaic, so they have all kind of different mutations in their germline. And what we do next is we cross these fish to wild type fish, and then we look at the embryos again. So again, after 24 hours, we do a hotshot DNA isolation, we do PCR, we check whether the mutation is there, and then we actually start sequencing the fish, the embryos that seem to have a mutation. And this means that these embryos are going to be heterozygous for an induced mutation, and they also have still a wild type allele because they have been crossed to wild type fish. And we basically sequence the heterozygous fish straight. We don't do anything special there. That means that if you get traces of sequencing, that you get complicated traces. And I've shown an example over here. So this is a sequencing trace of a wild type fish, and you can nicely see the sequencing trace. You can see the peaks occurring over here. However, if we sequence a fish that had a mutation, you get a complicated pattern. And there is actually software that promises to deconvolute these patterns and tell you what the two sequences were. I generally do not do this. I simply do this by hand because I find that it's just as quick. So what you do is you look at the sequences over here, and you line them up and you compare them directly and see what extra peaks have come up. And if you do this, you can see that over here there's an extra T. Here there is an extra T. Here there has come an extra C. Here there has come an extra G sequence because there's still a single G peak over here. So that means there has been, the shifted base is a G. Here you can see instead of a T, you can now see a T and a C. So there's an extra C has come up. If you now have a few of these sequences read, you can look at this sequence and try to find it back in the sequence over here. And as you can see, the TTCGC can be found back over here in this region over here. So it means that this particular sequence has shifted now to over here. And you can then count back. You need to shift this sequence 1, 2, 3, 4, 5 bases. So that would mean that this is a 5 base pair deletion in the sequence. So we know directly what is going on here. If this is too complicated, you can also go to the end of your sequence trace. Since we have very short PCR products, we actually read until the end of the PCR product, and you can get these, very particular, sequence endings in your sequencing traces. And since one allele will always be longer than the other when you sequence two alleles, which have a frame shift, it means that the messy sequence that you have over here at the very end of your PCR product, the sequence will clear up and will become clean again, because there's only one PCR product left that you're sequencing. And that means that you can actually find back patterns over here. So I see a green peak and a few red ones. And if you look at the earlier sequence over here, I can see the same pattern back. And you can simply move the sequence back and count back how many extra or how many bases have been inserted or deleted. So here you can see, I'm just going to change this. So here you can see an A peak, the last A peak, and then if you go back 1, 2, 3, 4, 5, you can see an A peak again that looks very similar. So you see the repeat of the pattern over here. So that means that we have a 5 base pair insertion or deletion. That's now not completely sure, but it's 5 base pairs, and that's really important because 5 base pairs means that there is a frame shift. So it's a frame shift of 5. It's not a multiple of 3, and that makes us happy because that means that, you know, this mutation will send the coding sequence out of frame. It's a frame shift mutation, and that will usually cause a null. So that's what we do. We sequence the progeny of such a cross, and then we raise that fish. We raise a parallel batch, or you can actually do tail clips and then identify the right fish, and then we raise these fish. We re-identify them. You know, we make sure that they have indeed the right mutation, and then we cross 2 of these different fish together to actually get a null phenotype. So that's how we make null mutations in the zebrafish. Now there are a number of other techniques, and I'm not sure how far I am. I still have some time. So there's a technique called Crispr, where you, rather than you know, making a mutation or making a null mutation, you try to block transcription. I must say that we have spent quite a bit of time with this, and we find that this is not very efficient. We have not had very much success with this. So I would, at the moment, I would not recommend using it unless there has been a new paper where they show that this technology is, you know, they have strongly improved this technology. Another technology is using crispins. This works quite well. So what you can do is you can, if you have a very efficient guide, is you can you know, identify mutants by directly injecting guides into the zebrafish embryo. Usually, if you inject a single guide, it is unlikely to suffice, and there's a statistical reason for this, and I don't have time to go into detail for this, but it means it's best, if you want to use this technology, is to select 2 to 3 different exons and design a CRISPR against every different exon, and then inject them together to create mutants, to create the crispins. So where you knock out the gene directly in the embryos that you are injecting. You have to be aware that if you want to publish such a method, that the reviewers might start asking, you know, it's very nice that you have crispins, but show us that you also can make a mutant that shows the same effect. So you have to be a bit careful, and I would always use these two approaches in parallel. So here you can see an example where I have created a double crispin. So we made a crispin for 2 different genes. This is an uninjected set of fish. Here we injected 2 patched 1 guides. They have a very slight phenotype. It's hard to see, but I can kind of pick it out. These fish were injected with 2 patched 2 guides. They're a bit more affected still. They look quite normal. But then if you inject a combination of 2 patched 1 and patched 2 guides, we create, you know, a double mutant, or here actually a double crispin for both patched 1 and patched 2. And you can see that these embryos are quite severely affected. And here you can see, for comparison, a real patched 1/2 mutant, and you can see that these embryos look pretty similar. So we did a pretty good job in knocking out both genes. Right. Now, another technique is gene editing, and this is actually something that many people are very keen on. So this is, rather than making a simple frame shift mutation, to create a null mutant. Rather, you want to make precise point mutations. And we have been using this, and we have had some success with this. It's still, it's still not super efficient, but it is something where you can really say, okay, this could be a good PhD project to start trying this. What you need to realize then is, how to do this. The first thing is, if you want to make a single point mutation, the guide that makes a cut in order to do this needs to be very close to where you want to make that particular mutation. It should not be further away than 10 bases of your target area. And I found this efficiency graph from the IDT website, I think, where they show this. So here they try to make mutations either on the cut site or further and further away from the guide cut site. And you can see that if you're very close, it's sufficient, but if you go more than 10 base pairs away, the efficiency drops off very quickly. Okay, so if you can only design or if you can only make a CRISPR about 10 base pairs away from your target nucleotide that you want to change, then it's you're very restrained in your CRISPR choice. You can only take a few CRISPRs will actually be near enough to the mutation that you want to make. Now, if this is the case, as I said, if you find a CRISPR nearby and it scores quite low, don't let these scores demoralize you. It might still work. If you really cannot find a CRISPR, then there are other systems. There, for instance, CPF1, where the PAM has a different recognition site. Maybe you can find a CPF target site near the nucleotide that you want to change. Of course, if you have very few choices for your CRISPRs, it might not be possible to find the restriction site that gets, destroyed or damaged by an active CRISPR-Cas9 complex. So that means that you need to have a different test to see whether your CRISPR is actually active. And what we use there is a very simple test. We call it a smear test. It's very low tech. The only thing that we do is we inject our fish with a CRISPR, and then we make a very short PCR product. So again, we try to get it about 120 base pairs. That is really, really short. And then we compare injected embryos with uninjected embryos. And you can see an uninjected embryo over here, and your PCR product is really nice and sharp. You get a crisp band. However, if you look at the injected embryos, you can see that the bands are all very smeary and hazy. And that is because the CRISPR is working. It's creating all kind of, base insertions and deletions, and that will cause, PCR sizes to be different. So you get a very, imprecise PCR product that is, it's covering a range of sizes. So that means that we know, yes, the CRISPR, the guide that we're using is efficient and is working. If you don't want to use this technology, there's another technology called CRISPR-Stat, and I've put the website for that over here. Okay, so if you want to make such point mutations, you need to make, I didn't say this, you need to create basically a primer or a piece of DNA that will guide the mutation that you have to make. So what we use to actually, change the sequence is we use 127 base pairs, roughly, editing oligos. These editing oligos have the edits that we want to have in the DNA. In addition, these oligos are phosphorothioated because they are more stable. And actually, IDT appears to have a new modification, and you may want to try that. Maybe they're even better. They're called Alt-R modifications. So we make these oligos in a particular way, and I've tried to show this in this figure over here. So these oligos are asymmetric, and they're complementary to the non-target strand. Okay, so I show this over here. So the oligo goes in this direction. If we say that we want to make the edit over here, then there should be 42 base pairs on this side, and there should be 85 base pairs on that side. And this is the paper that basically says these oligos are the most efficient. And this is only for Cas9. If you use another enzyme, you need to design your oligos in a different way. Okay, so once we have identified where we want to make the oligo, we need to do additional things. And this is really what I call the puzzle, because it's really complicated. But we usually get it worked out. So first of all, we want to add additional wobble bases. We want to add further wobble bases in the coding sequence to ensure that we create a primer binding site, and we're going to order a corresponding primer then that will only bind the target area if the edit has been made. So this is a kind of our positive control that we're going to use to see whether the edit that we're trying to make is actually going to be made in the genome. And for that we need another primer, and this primer needs to be outside of this targeting oligo. Otherwise we simply start amplifying the oligo that we co-inject. When we design these, when we change these wobble bases, we keep an eye on coding frequency. I've put a link over here. What we also do is we edit in such a way that we try to create a new restriction site. And again, if we try to create a new restriction site, we use enzymes that cut and PCR mix to make our life easy. Finally, we need to do one more thing. We need to ensure that when the edit has been, entered in the genome, that the guide doesn't keep on cutting the edited genome, because otherwise you will create additional mutations that you do not want. So we edit in such a way that we, for instance, destroy the PAM site, that there's no cutting after the first cut has been made and the edit has been done. Then the guide should not work on the genome anymore. So here's an example for a particular mutation that we're trying to make. I actually do this old-fashioned and do this in Word. I have a whole load of different colors over here. So if we want to make a D2998A mutation, so we change the D to an alanine, then this is the codon that we want to change. If we want to change to an alanine, we have a number of options. We have a GCT, GCC, GCA, or GCG, and this is the codon frequency. This one is very rare, so I'd rather not make that particular change in the genome. What you can see in yellow is the guide sequence, and in gray you can see the PAM site, but it's reverse complement. So it's not NGG, but it's CCN. Then in bold you can see the target codon, as I said, and we have some alternative bases over here for wobble sites. And you can see that the ones in red I'd rather not use, but I will use them as I have to. Then this particular nucleotide is very close to a splice acceptor site. I'd rather not change any nucleotides that are close to this, that are part of this splice sequence. But outside the splice sequence, I can make a few changes. And here I edited these three bases over here, because this is not coding sequence. I can basically change it to whatever I want. Right. Okay. So then I simply I try different versions of changing these nucleotides, and then I see if primer 3 is capable of identifying a primer that will bind the mutated sequence. So here we have the modified sequence when I made all the changes. And if you look closely over here, you can see a TCT-AGA. So this has introduced a new XBA1 restriction site in just, outside the coding sequence in the intron. And we can use this to identify, the edited allele. I also have identified a primer binding site that will bind over here. It's underlined over here. And you can see that this primer will have a lot of mutations compared to the original sequence. So all the mutated bases are shown in green over here. And this will then allow us to design 2 primers that can amplify only if the edit has happened in the genome. So what are we injecting? So we inject the tracer, the guide. IDT says it's good to preform these molecules. So you keep them for a few minutes at 95 degrees and let them cool down. Then you add the Cas9 protein. You add some phenol red. That's simply a tracer to show where you've injected. And then you keep them at 37 for a few minutes. Then when the active CRISPR-Cas9 complex has formed, you add the editing primer. And this is then what we inject into the zebrafish again. And then we do a PCR test for the editing after 24 hours. And this is what you hopefully should get. So here are embryos that have been injected with the editing mix. This is an embryo that has not been injected with the editing mix, and this is just a water control. And you can see that only the embryos that have been injected with the editing mix, will allow amplification with the edit-specific primer. We can then also do, we can use primers that flank the editing site and then use the new restriction enzyme to get an idea, are we inducing any new restriction sites? And here we have just a whole load of embryos that have been injected. And yes, we see a few embryos over here where suddenly we get a smaller product after we cut with this enzyme. So there seems to be some editing going on here, and that's very encouraging. What we do then is we raise injected embryos. These embryos are mosaic, so then we will have to identify, we have to cross the G0s and then look at individual embryos that are progeny from such a fish to see whether we find any fish that have the edit. And it can be seen over here. So here we have a panel of F1 embryos from a particular founder fish. And you can see that 3 of these embryos, when we use the edit specific primer, we see 3 embryos actually, yes, they get an amplification. And if we see that, we can then take those particular embryos, we then amplify with the flanking primers again, and then we do sequencing of the DNA. This DNA is then again going to be heterozygous because we crossed our G0 fish to a wild type fish. So we have a wild type chromosome and we have a mutant chromosome. And you can see a sequence trace over here where you can very nicely see that, in 2 bases where we wanted to have the edit, we can suddenly see we have double peaks. So that means that the fish are heterozygous for the mutation that we want. And this is how we, are currently now, creating very specific disease models where we, choose exactly one amino acid that we want to change into another amino acid, and then we make that mutation in the zebrafish. Okay, I think I'm getting close to the end of the seminar. I'm going to skip this. If you want to make more, larger insertions, larger changes of the genome, we haven't used this at the moment. We're gearing up to this. If you want to do this, there's a very good online protocol. It's shown over here. I'm just going to get the thing out of the way. So this publication over here gives you a very detailed protocol on how you might do this and I can truly recommend this. We're actually gearing up for this now. I'm going to stop here. I just want to acknowledge the people who have been working on this, and I'm ready to take questions now. So I'm not sure what I need to do now. Yes.
You just sit there and I will read out the questions. So
okay.
Thank you very much, Beck, for really nice. It's very nice to see such a kind of detailed idea about actually how to do it, and I hope it's been interesting to the audience. So as a reminder, please just go ahead and type questions into the box that you should be able to see on the GoToWebinar control panel, and I will read them out for Frank to answer. So as a starting point, I guess I was interested in what sort of proportion of HDR events versus sort of background NHEJ deletion type things do you get when you try to make those point mutations?
Okay, so that is very oligo and CRISPR dependent.
Mm-hmm.
So I think we need to raise about we once had some statistic. We need to raise about 36 G0s to get a transmitter. That sounds like a lot, but it's for fish, you know, that's one tank.
Yeah.
So in that sense, it's quite doable. But it's so, and this is an average. So some of them, you know, within 10 fish, you will have one that transmits the allele that you want. But in other ones, it was, you know, 60 fish before people found the fish that they wanted. So it is, and we think it is dependent on, you know, how efficient your guide is. Mm-hmm. So it's really important to test, you know, how well is my guide actually making mutations. If it's very poor at that, then I would probably not advise using it. But mm-hmm. There's probably also factor X, which is, you know, the oligo that you're using. Some oligos we find are very efficient. Other oligos we don't, you know, they're not very efficient, and we don't get the statistics there. We don't have the money to try 100 oligos and see, you know, what the sequence requirements for this are. So we generally, you know, just try injecting. And as I said, if you of course, you can do these very quick tests after 24 hours after you've injected your oligo and CRISPR guide combination. You can do your PCRs. If you see a lot of bands coming up that were not present in your uninjected embryos, then you know, yes, something is happening and it's probably okay.
Mm-hmm. Yeah.
If you don't see that, if you see a lot of negatives in your injected embryos where you don't see any amplification, then I would say, yeah, probably you know, try something else. Try another guide or try another oligo to make your mutations. But I can say that we have had at least 5 successes now in the department where people did that, and it's maybe out of trying 11 or so. So it's about 50% success rate. And usually you can discard the unsuccessful ones quite quickly because they don't give these PCR. So there's not much time lost. It's not that you have to raise, you know, the unsuccessful ones were usually weeded out in the very early stages, and not, you know, after 3 months raising adults and trying to find a mutant and then finding that it didn't work. So the cost is quite limited in that respect.
Sure, yeah. So it's not like you have a particularly strong feeling of whether HDR is more or less relatively efficient than it might be in a different organism. It's the same sort of principle of you just need to do it on a fairly large scale. And
it's I think you know, and what's it called, non-homologous end joining is probably more efficient. I'm quite sure of that.
Yeah.
So it's probably the, you know, it's 20%, I would say. You know, of the alleles will be you know, driven by the oligo, and 80% will be simply insertion deletions. So I think that would be realistic. Unfortunately, there are also some hybrid things. So sometimes you get your mutation, but then you also get an insertion or a deletion, and that's of course not what you want. So even if you edit your allele such that the guide stops working, you will get a few dirty alleles, so to say.
Sure, yeah. So we've got 3 questions in the chat at the moment. The first one is from Radha Kulkarni, and she says, thank you for the talk. You said that the chop scores did not reflect the guide efficiency. In this case, how do we select guides for crisp ants that need very efficient guides?
Okay, so the good news is many guides work very well if you simply use them by ordering the RNA and the protein. 75% of the guides work, if you randomly pick a guide, even if you don't even look at the scores at all, I would say 75% work to a very decent degree. So you could even simply go and say, I'm just gonna, look for a GG somewhere, and then I'll take it from there. So the only thing that a guide needs is a PAM site, right? So if you order in protein, if you order in the RNA and not make it yourself, it is very easy to get an efficient guide. It's really not hard. So I would still have I would look at the scores, but I would also say, okay, I'd rather have one guide in exon 1, one guide in exon 2, and one guide in exon 3, and I would simply take the best scoring. Or if you say I really want to see whether they're working, then pick one that has a restriction site close to where the guide actually cuts, and that it will lead to destruction of the restriction site.
Sure. Second question from Mael Belek. It says, have you tried the new Cas variants SPG and SPRY with different PAM by any chance?
No, we haven't tried. No, we haven't tried. So we haven't really had the need so far. If you really want to make one particular mutation and there's no guide near, then you kind of think, okay, I should take another one. But we never had that issue. We always found a guide that we needed, pretty close to the mutation that we wanted. And as I said, there's CPF1, which has another specificity, which is very T rich. So GG and CC are by default, if you have an AT rich region, then that's not gonna, you don't find very many. But then you have CPF1, which has the TTTV PAM site, which will work quite easily. So and we've tried that one, and that works pretty well as well. So I'm quite happy with that. The new variants, I'm not sure how easy it is to get them, because you probably would have to. I'm not sure whether they're commercially available as proteins.
Yeah. So I guess the big advantage of Cas9 is you can just buy it.
Yeah.
Have you tried different suppliers for Cas9 actually and found any variability in that?
So we have tried IDT and NEB, and both of them work fine. So we go by price basically, and laziness, because we know that NEB works. So and they have it in micromolar, and it's easy for our calculations. So we tended to have NEB for our and we have very good experience with that. NEB is a good company, so that's.
We've got a question in 2 parts from Antonio Martinez Silva. The first part says, thank you so much for the tips. I'm actually genotyping my F1 and could be difficult to find the changes in the sequence with respect to wild type. And the second part says, could you talk about software in development to analyze the sequences?
Okay, so software that deconvolutes. Yeah, as I said, my neighbor next door uses it, and I had just looked at some sequences this morning, and I felt like I can just as easy and look at it. So I haven't really, but if he can supply, or he or she can supply an email address to me, I'm easy to find online. Just type in my name and you will find me. Send me an email, and I can inquire next door to see what kind of software they're using, because it looked pretty nice.
Thank you, that's really helpful. Another question from Ana Lopez Ramirez. How do you see qPCR with primers targeting the last exons in the gene to evaluate RNA nonsense mediated decay of the specific gene after CRISPR injection?
We haven't really done much qPCR CRISPR injected ones, we usually then go through the genome and, go through the generations and generate the mutants before we do any qPCR. And then the options are, you can use basically any qPCR primer set to do that. And we usually use GenScript to design. They have a website where you can kind of design your own PCR primers.
Yeah. I've just had a comment here. I guess people online can't see the question, so I'll just read this out. Jordan Walker says that Crisp ID is a good deconvoluter. So there's a recommendation from Antonio. Two more quick questions, and then we better wrap it up. So one of them's from Anantha Krishna Tantri, and says, have you tried CRISPance? If yes, what was the efficiency you found?
So making CRISPance works pretty well, but it's best to pre-select your guide. So when we do this, we order 4. So we pick 4 using ChopChop, and we spread them out over different exons ideally, and then we inject all 4 of them, and we do these quick smear tests. And then we look at the guides that give us good smearing. And then we pool usually 2 or 3 of those to do the final injection. And that works really, really well, surprisingly well, much better than Morpholinos, which are really expensive and have a lot of issues. So if you tell me, what do you want to use, Morpholino or CRISPRs, I use CRISPant all the time, definitely.
And then one final very quick question before I wrap things up from Swati Radha. Thank you for the great talk. Have you tried making cell-specific knockouts?
No. This has been on our mind for a long time, but it requires you to generate transgenics and specific. But it's definitely not impossible. It's definitely possible, but it will take a serious amount of time to get this working.
Yeah. Brilliant. So I better wrap things up now as we're almost at time. So thanks very much indeed to everyone who's come today and for the questions in the chat. And thanks very much to Frank for a really, really interesting talk and for all your responses to the questions.
Yeah, I'm happy to answer questions by email. If anybody didn't get it, or I went too fast, I often go too fast, or it was too basic or too advanced, then just send me an email and I'll try to respond.
That's fantastic. Yeah, thank you for that. I've also been reminded to tell you that you can continue the conversation online following at BiochemSoc or at PP Publishing. We welcome suggestions for future topics and speakers to feature in this Biochemistry Focus webinar series. If you have an idea for a webinar in 2023, we invite you to submit a proposal for an upcoming webinar. You can find more information about the webinars, propose your webinar, and watch previous recordings on www.biochemistry.org/events-and-training. Just go to the website and have a look. Please do look on our website for all upcoming future webinars, but if you have missed any of the 50+ webinars that we've run as part of the series or would like to watch them again, please visit our website or YouTube channel. So details should be shown on the screen there, hopefully. The recording from today's webinar will also be available to watch within the next couple of weeks. Join us on 10th of March at 1400 GMT for our next webinar, Transitioning Between Academia and Industry and Vice Versa, where we will hear from 4 speakers as they discuss their individual career paths and motivations, as well as provide helpful tips and advice on the skills needed during the transitioning process. Finally, I'd like to highlight that it's more important than ever to stay connected and engaged with your fellow molecular bioscientists. It's an exciting time to join the Biochemical Society's community of researchers and specialists to stay connected and take advantage of key benefits, including discounted registration fees for society courses and meetings, exclusive access to a wide range of grants and bursaries, personal online access to 2 of their journals, and more. Visit the website to find out more. And finally, I think that's all I have to say today. So thanks very much again to everyone for coming, and thanks to Frank for a really, really interesting webinar, and goodbye, everybody.
Bye.
Bye.